पूछो जाके उनसे जिन पे रोशनी नहीं, पूछो जाके उनसे कैसी लगती है कमी.
Good evening, people. Today's session's topic, I guess everyone is aware that anatomy and physiology of cornea, which will be presented by Aashna Khan, who actually is my junior. She is a first year optometry student from Jamia Hamdard. She herself is a masterpiece, so I think that's ample for her. So without wasting time, let's get started. Over to you.
Okay, thank you so much. So today we will have an overview of the ocular structure, which is which is also referred to as the window of our eye, and that's the cornea. So let's begin. Talking about the anatomy of cornea, the cornea is a transparent avascular structure. It forms the 1/6 of the outer fibrous coat of the eyeball. We all know that the eyeball is consists of three coats, that is the fibrous, vascular, and nervous. The fibrous being the outermost, and the nervous being the innermost. So the fibrous layer consists of the cornea and sclera, of which the cornea forms the 1/6 part, that is the anterior 1/6 part, and the rest 5/6 part is formed by the sclera, which is the posterior most part. The refractive power of cornea is about 43 diopters, which is 3/4 of the total refractive power of the eye, which is 60 diopters. The total refractive power of our eye is 60 diopters, out of which the cornea comprises of the 3/4, that is 75% of the total refractive power, and it is 43 diopters. The rest, that is around 17 to 20 diopters, is comprised by our lens. And the cornea is the main refractive surface of the eye. It forms the major refractive surface of the eye. How we will get to know in the later slide. So dimensions of the cornea anteriorly and posteriorly are: the anterior surface is elliptical, that means it is oval, so it will have both the horizontal and vertical diameter, out of which the horizontal diameter is 11.75 mm and vertical diameter is 11 mm. You can see it in the picture that the anterior surface of the cornea has two different diameters, horizontally and vertically. The posterior surface is circular, so it has the same diameter in all the angles, which is 11.5 mm. The thickness of the cornea also varies from center to the periphery, which is 0.52 mm at the center, and at periphery it is 0.67 mm. It is thicker at the periphery than the center. The radius of the curvature here, central part of cornea is referred to, and the anterior curvature of the cornea is 7.8 mm, and the posterior is about 6.5 mm. Moving on to the next slide, we have histology. That means the study of the structure of the cornea or the tissue layers. Talking about anteriorly to posteriorly, the cornea is considered to consist of following 6 layers, that is the epithelium being the outermost, then the Bowman's membrane, then comes the corneal stroma, which is also referred to as the substantia propria, then the pre-Descemet's membrane, and lastly the endothelium, which is the innermost layer. You can see in the picture that the outermost layer is the epithelium, then comes the Bowman's layer, then the stroma of the cornea, that is also known as the substantia propria. Then comes the pre-Descemet's membrane. Here it is not mentioned in the diagram since it is recently discovered in 2013. After that comes the Descemet's membrane and the innermost layer, which is the endothelium. We'll read about these layers one by one. So our first layer is the epithelium. The epithelium of cornea is stratified squamous non-keratinized type. That means it does not consist of keratin, so that what it makes the cornea as a living cell. It is continuous with the bulbar conjunctiva epithelium at the limbus. As you all know that the limbus connects the cornea and the sclera. So if both are connected, then the epithelium will also be connected to each other. That is the corneal epithelium is also connected with the bulbar conjunctival epithelium. Bulbar conjunctiva is present in the sclera, so they both are connected to each other. So the epithelium represents about 10% of the corneal thickness, and it consists of 5 to 7 layers of cells, which is divided into three layer types. The deepest, that is the basal layer, the mid epithelial layer, and the superficial layer. You can see in the diagram that the basal layer, which is the deepest layer, consists of generally one layer of cell, and it is the columnar cells. They are tall and column-like. Next is the mid epithelial cells, which are known as wing or umbrella cells, and they are present in 2 to 3 layers. Okay. And the superficial, that is the most outermost part of the epithelium, is composed of flattened cells, which are present in 2 layers. And these cells, the superficial cells, also consist of microvilli at the top. The corneal epithelium sheds at a regular interval and is replaced by growth from its basal cells via mitotic division. The entire epithelium is replaced in a period of 6 to 8 days. So what it means that when the corneal epithelium is removed or it is shed off, then the basal layer, which consists of the basal cells, columnar cells, they undergo mitotic division, and then the cells, the new dorsal cells that are produced through the division, mitotic division, they migrate and they will already move to the epithelium. About the nerve supply, where I said that these are supplied by the long ciliary nerves, which come from the ciliary body of our eye, and they are the divisions of the nasociliary nerve, which itself is the ophthalmic division of the trigeminal nerve. So when the long ciliary nerves combines with the conjunctival nerves, they innervate the whole of the cornea, forming the pericorneal plexus. That means a group of nerves, sort of. And out of those pericorneal plexus, around 70 to 80 nerves innervates the stroma of the cornea, forming the stromal plexus. And when they do so, the corneal nerves lose their myelin sheath. After that, after innervating the stroma, the nerves come to the sub-epithelial layer, and there they form the sub-epithelial plexus. That is the group of nerves also innervates the epithelium and forms a meshwork of nerves. And there, when they in this way they supply whole of the epithelial layers of the cornea, and at the last they also lose the Schwann sheath. No nerves in the central posterior part of the cornea, desmoid membrane and endothelium. So except these layers, all of the corneal parts are very rich of nerve supply. You can see in the picture that nasociliary nerve का division है, short posterior ciliary nerves and long posterior ciliary nerves, out of which the long posterior ciliary nerve supplies the nerves to the cornea after further dividing. Now coming to the most important part, that is the corneal physiology. So how the cornea functions. The cornea performs the following functions by maintaining its transparency and displacement of its tissues. The cornea acts as a powerful refracting lens of fixed focus. Since we know that the epithelium and all of the layers of cornea are very uniformly arranged, so it forms a very uniform surface, and it is also moist. So it acts as a powerful refracting surface of the eye. The cornea also protects our intraocular contents. The barrier functions helps fluid loss and pathological entrance to the organism. As I've already said that, why it prevents the fluid loss. Since it acts as a barrier, so the fluid loss is prevented, and also the pathological organisms are prevented from entering the eye. The cornea also helps in the absorption of topically applied drugs. The drugs which are applied topically over the eye, the cornea helps in their absorption also. It serves as a major process to respond to wound healing after anterior segment surgery. So whenever any anterior segment surgery of the cornea, like the person suffers from any sort of trauma, so it's not a major issue, since the basal layer of the cornea, which is comprised of the basal cells, columnar cells, they continuously divide and produce dorsal cells which migrate to the upper surface of the cornea, the epithelium layer. So it is continuously regenerated. So that's a very important function of the cornea. Moving further, we have a question that why is the cornea transparent? Maintenance of high degree of corneal transparency is a prerequisite to maintain the main physiologic function of cornea, that is to act as a major refractive medium. So, the corneal transparency is very important to let the cornea perform main physiologic function, that is to act as a refracting surface, as a proper refracting medium. So, and this is a result of various anatomical and physical factors, where the anatomical factors constitutes of the corneal epithelium and tear film, which is, as I've said, that very regularly and uniformly arranged in the epithelium, consists of junctions and is very compact, and also the tear film. Maintain the homogeneity of the refractive index, since it keeps the corneal surface moist. So in that way, the refractive index is also maintained, and hence the cornea becomes transparent. The arrangement of stromal lamellae is also one of the most important points, as we have seen that the stromal layers are very parallelly arranged to each other, and also they are parallel to the whole corneal plane. So this is also a major transparent. And the corneal avascularity. The cornea, as I said, does not have its own blood vessels, and this also plays a role in maintaining the transparency of cornea. We have a physiological factor which affects the corneal transparency as corneal dehydration. The water content of normal cornea is approximately 80%, which is the highest of any connective tissue in the body, and is kept constant by a balance of various factors. Disturbance of any of these factors lead to corneal edema, where the hydration exceeds 80%, central thickness increases, and transparency reduces. So we can say that the thickness of cornea and the hydration are directly linked with each other. How? Because if the water content will increase, then obviously the cornea will also become thicker. So, and when it happens, when the hydration exceeds 80% of the water content, and it leads to corneal edema, that is the swelling of the cornea. And when it happens, then obviously the transparency of the cornea will also be reduced. Now I would like to share a few facts or questions type which I have collected. So our first question is, what is the logic behind not accepting cornea from a live donor? See, if we accept the cornea from any live donor, then the person will not be able to see properly, as the binocular function of the human eyes will be lost, which is maintained only if we have two eyes. And also the stereopsis will also be lost. The person may get affected in viewing he or she will not be able to view the 3D images, and therefore it is not accepted from a live donor. The cornea is basically collected after death, between 4 to 6 hours of death, not when the person is alive. When we talk about kidneys, they do not function simultaneously. That means if one of the kidneys removed, then it will not be a major issue, unlike the human eye. So a kidney can be donated when the person is alive, and even the liver can be transplanted, but it is not in the case of eye, and that's what it makes it unique structure. Our next question is, why the cornea and the lens of the birds transmits ultraviolet light while our cornea and lens blocks it? So as we know that UV light wavelengths slightly shorter than those visible to humans. We see the visible light of the electromagnetic spectrum, that is the Vibgyor. We are not able to see the UV light, since it's very powerful, and it's not good for our eyes also. The UV light is basically perceived by few of the arthropods or chordates, but not by the human beings. So most birds can see UV light. This capacity is possible because birds have a fourth type of cone that is sensitive to this wavelength, and because the ocular media allows this light to enter the eye. Human eye has three types of cone, that is the red, green, and blue, but it is not in the case of birds. In few of the species of birds, where they also have a fourth type of cone, which is specifically present to capture the UV wavelength of the eye. Thus, the presence of class of photoreceptor, which is maximally sensitive to UV light, which is the fourth cone that I talked about, depending on the species, is the reason behind it. The roles of UV in vision are manifold for the birds, not for us. From guiding navigation and orientation behavior to detecting food and potential predators, and to support high-level tasks such as interspecific communication and all. So that is the reason why the lens and the cornea of the birds or other chordates or vertebrates, except the human being, transmits ultraviolet light, but our cornea doesn't. So these are the references where I have visited and taken help to make this presentation, and that was all about it. Thank you so much.